Hello and welcome to the fifth of a series of video lessons in pharmacology. As I've said before, to get the most out of these lessons, I strongly suggest you watch them in order, beginning with Lesson 1, an introduction to pharmacology. In earlier videos, we were introduced to catecholamines and cardiac beta-adrenergic receptors. We also examined how calcium within cardiac myocytes is able to regulate muscle contraction. We did not, however, discuss how processes occurring outside the cell are able to control what happens within the cell. Addressing this question is the goal of this video, and we will begin by mentioning the work of two scientists. Walter Cannon was a physiologist at Harvard University who in 1915 coined the phrase fight or flight, a term we now use to describe the acute stress response. His work demonstrated that the acute stress response is mediated by epinephrine that is released from the adrenal gland. We will also briefly discuss the work of Earl Sutherland, a biochemist at Vanderbilt University who in 1971 won the Nobel Prize in Medicine and Physiology for demonstrating that the mechanism of action of epinephrine and other hormones involves cyclic AMP, which he called a second messenger. Sutherland's work began with studies of liver glycogen metabolism. However, we will consider how his discoveries pertain to the heart. You are aware that if you see, smell, taste, hear, or feel something frightening, then your heart will often beat faster and harder. Imagine you are a scientist trying to understand this process. How would you do it? Walter Cannon devised an animal model in which he could study the cardiac effects of the acute stress response. When he subjected animals to a stressor, as you would expect, he observed an increase in heart rate. Cannon then surgically separated the nerves going to the heart and observed that the heart rate still increased in response to a stressor. He inferred that whatever was causing this response was reaching the heart by the circulation. In a third group of animals, he then denervated the heart and also surgically removed the adrenal glands. In these animals, the heart rate response to the stressor was abolished. By the 1930s, Cannon concluded from these and other studies that the acute stress response was mediated by epinephrine produced and released from the adrenal gland. We now know that the fight or flight response begins in the cerebral cortex and that neural signals travel to the amygdala, which has a primary role in processing emotional response. These neural signals then travel to the hypothalamus, brainstem, spinal cord, pass through the celiac ganglia, then to the adrenal gland. The adrenal gland then releases a chemical signal, epinephrine, that circulates throughout the body. After reaching the heart, epinephrine binds to beta-adrenergic receptors and causes the heart to beat more forcefully and more rapidly. The last piece of this puzzle was understanding what exactly happens after epinephrine binds a cardiac beta-adrenergic receptor. This brings us to the work of Earl Sutherland. Sutherland discovered that after epinephrine binds to the beta-adrenergic receptor, abbreviated BAR, an intracellular enzyme produces a chemical compound called cyclic AMP. Epinephrine is the first chemical messenger. Cyclic AMP is the second messenger. Adenosine triphosphate, ATP, an organic compound found in all forms of life, provides the energy to drive this process. The enzyme adenylyl cyclase converts adenosine triphosphate to cyclic adenosine monophosphate. Cyclic AMP then activates other enzymes that are responsible for chemically altering calcium channels, resulting in more calcium able to enter the cell and thereby enhancing muscle contraction. Sutherland did not know exactly how the beta-adrenergic receptor was linked to adenylyl cyclase. In the 1970s, Alfred Gilman and Martin Rodbell discovered that the beta-adrenergic receptor is coupled to adenylyl cyclase by proteins that they named G proteins. In 1994, Gilman and Rodbell shared the Nobel Prize in Medicine and Physiology for their discovery of G proteins. We now say that the cardiac beta-adrenergic receptor is a G protein coupled receptor. G proteins bind GTP, guanosine triphosphate, and so they are also known as guanine nucleotide binding proteins. The subscript S indicates this is a stimulatory protein. Therefore, epinephrine binds to the beta-adrenergic receptor. This causes the receptor to activate a stimulatory G protein, which in turn activates the enzyme adenylyl cyclase. Once the signal enters the cell, elevated levels of cyclic AMP in the sarcoplasm activate protein kinase A. This enzyme phosphorylates plasma membrane calcium channels, increasing calcium entry into the cell. Protein kinase A also phosphorylates sarcoplasmic reticulum calcium channels, further enhancing intracellular calcium release. The key point is to understand that as intracellular cyclic AMP levels rise, intracellular calcium levels also rise. The last point to consider is what happens to cyclic AMP. After all, this would not be an efficient system if cyclic AMP levels remained elevated. Cyclic AMP has a high-energy phosphodiester bond. The enzyme phosphodiesterase breaks this phospodiester bond, converting cyclic adenosine monophosphate to adenosine monophosphate. Milrinone, amrinone, and cilostazol are 3 drugs that inhibit cardiac phosphodiesterase. We refer to this group of drugs as PDE inhibitors. PDE inhibitors produce positive inotropic and chronotropic effects by inhibiting breakdown of cyclic AMP, which, as we've just seen, is associated with increased levels of intracellular calcium. For clinicians desiring to increase intracellular cyclic AMP levels, a more direct treatment is to administer a beta-adrenergic agonist. There are numerous beta-adrenergic agonists available. I've listed a few of the more important cardiac drugs here. We've previously discussed isoproterenol, epinephrine, and norepinephrine. I've also included dobutamine, a synthetic beta-adrenergic agonist that is commonly used to treat heart failure and shock. Let's conclude by summarizing a bit of what we've covered so far. Patients who are hypocalcemic also have low levels of intracellular calcium. Severe hypocalcemia may cause arrhythmias and heart failure and may be treated with intravenous calcium in the form of calcium gluconate or calcium chloride salts. Recall that the cardiac calcium channel is a voltage-gated L-type channel. In the previous video, I also very briefly mentioned a group of drugs known as calcium channel blockers. These agents reduce intracellular calcium levels by inhibiting the movement of calcium ions through the selectively permeable membrane calcium channel. We will discuss calcium channel blockers in more detail in a future lesson. Now, in contrast, beta-adrenergic agonists and PDE inhibitors increase the flow of calcium through calcium channels. In the case of both of these drug groups, this action is mediated by cyclic AMP. Before leaving you with an illustration of the chemical structures of ATP, cyclic AMP, and AMP, I want to remind you that if you would like to see more videos, please subscribe to this channel and also give these videos a thumbs up. Your support in this way is vital for the continuation of this channel. Feel free to leave suggestions, questions, and comments below, and as promised, here's our last figure. Thank you.